Doordarshan Manipurna Sandukliba Program on Education Gamari Lena, Akogi Manipur University Department of Biotechnology da Kuna Ladiba Wazasing, Adeki Maru and faculty members, Amasung Mahiroi Singer, Hariba, Kun Department Sita Karikaramba, Kogi Karikarma Tabaxing Honoribagi, Hibodokoi, Mayam, Koi Amutang, Punami Taminasi. Department Asigi Head Oibiriba Professor R. Paratharajanna Department Asigi Matangda Wapam Kaya Tambiraki.
Biotechnology department was started way back 2006. The department is extending a lot of work in the field of basic biotechnological approach required for the state and through the education of students in different fields of biotechnology. This department offers a 2-year post-graduation course as well as PhD degree in biotechnology. And we have a good number of teaching faculties. They are all well trained. 1 professor and 5 assistant professor and 5 guest faculties are available. They are all having good background in biotechnology and some of them are trained in abroad also. And every year in our department we take 22 students in our per year. And all these 22 students are given proper training, hands-on training, both in practicals of different fields. And our department has produced 10 PhDs so far. And at present, nearly about 2 dozen scholars have registered for PhD program. And I am sure that more and more scholars will be coming. And the department is getting a good amount of money from different funding agencies like UGC, then CSIR, then DBT, DST, ICMR, the Indian Council of Medical Research, like that. And the department has basic research objective like cancer research and molecular biology, and then clonal propagation of bamboos, then orchids and certain medicinal plants, because medicinal plants are becoming gradually dwindled day by day. So the important plants which are threatened, these plants are collected and then through tissue culture our scientists are trying to propagate them so that they are available in large numbers. And some research scholars are working with the protein research as a main area. And evolution and transmission of drug resistance among the people infected with TB, tuberculosis. Tuberculosis, that is why again another important area. Then DNA methylation analysis and drug dependent population of Manipur. These are all some of the primary objectives of the department which we have been doing it for the last 1.5 decade. And so far a number of students have come out with glorious degrees and approaches, but out of them a quite good number of our students, you call them as alumni, they are all well settled, well placed in different parts of the country and also abroad. So we have more than 30-35 people who have given their address, because many people we could not contact because they go abroad and then settle down. But we gathered some information. So nearly about 33-35 students have given their name and where are they working, what is their position, designation, type of works they are involved. All these details they have given. And when in the MSc Biotechnology, the opportunities for students is again enormous.
Department Asigi Oina, Sahi Anigi post graduation course Kidamak Satra Masing Kungani Twegi intake capacity lay. Masikusi Matakta PhD degree Tawagi Kudong Sabusu Piri. Department Asida Professor Ama, Assistant Professor Manga Amadi, Guest Faculty Mangana, Punna Department Asida Takpi Tambiri.
Potentially severe infectious disease caused by a bacterium called Mycobacterium tuberculosis. According to World Health Organization, TB is the 13th leading cause of death and 2nd leading infectious disease after COVID-19. The ABCC1 gene, which is located on chromosome number 7, which has 28 translated exons encoding a protein of 1218 amino acids. So the ABCB1 gene encoded by P-glycoprotein has played an important role in pharmacokinetics and transporting toxic metabolites out of the cell. So here are some of the methods I have followed in doing the project. So I collect a sample of about 40 samples regarding my project. So DNA is extracted, then we have run the agarose gel electrophoresis, and quantification of DNA is done with the help of spectrophotometer. Lastly, the genotyping of the DNA is done by using qPCR, using Tecman allelic discrimination, and the result was statistically analyzed.
Department asida biotechnology gi field kayada tongan funding agency singi makhada research project kaya ama lepa leitana paikhatli. Project kaya sing asigi matangda project asibu paibiriba oza singna project singi oiba nungi wakalon asumna thambirakhi. Akhoi new zealand university akhoi Dr. Nongdam, assistant professor, Department of Biotechnology, Manipur University. Ahanbada ozabu Doordarshan Manipur mai taram seri.
Seri.
Ahanbasi ozab isagi field se karma biotech to oidin sir.
Aigi se basically aina tawdari se plant biotech ke field ni aduga aina zik maruina tawdari se khoina khogi aigi line haitaren area of research site betare. Masida ain tawdari se khogi conservation genetics study aduga in vitro plant propagation. So sigi area maruina khoina focus tawjari ji sigi area sidani hek tawjari si aduga khoina hojik keno si aigi lab se tawjari si aina sai hojik haijagi ba conservation genetics harise masi yamna hojik ki matam gadi am suna ba haitare haidi conservation gi khogi conservation plant genetic resources mangdar iba deplete tawthare iba khogi society sida khogi environment impact mayam leibadagi khogi resources mayam handare masi conserve tawnaba khoina masi kari tawwe age haitaragadi khoina khogi conservation genetics sida khoin tawrise khogi conservation status khogi plant sigi adi genetic status am yengba taka oi moije kallibra moije solabra haiba sima khoi yengthokpa ma change ada khoina khoigi hujiki genetic status conservation status khoi khangtok pasi khoina tawdui si khoina genetic diversity of the plant gi genetic diversity khoina khangtok bata asi khoina molecular marker season dana molecular mechanism season dana khoina khoigi plant sigi particular plant sigi natural plant resource sigi khoigi masi sondari de masi kandana leiri haibase khoi dum khangba ngamni haitare akoi genetic diversity study se tawraga aduna khoina kanba sonba haibase plant sum akoina layman tam dani ko aina haijari je ko aja haiba haitare ga khoina kanba plant si genetic diversity ji wanglera ma khoina sari taw matam sida atadi moise kalli haina lowe they are strong haibi ai kaigino haitare ga moise khoigi level of genetic diversity is very important akoina moigi ngasagi plants ina khoigi environmental factors environmental impact sida mayam pisala pasida khoina adjust to ngamba haibase ngamna ba haibase moigi diversity level to wang khat pada lay moi diversity level to wang targadi moi adjust to ngam doine survive to ngam doine adjust to ngam doine adu diversity ne low targadi hoi sole haiba mukti haiba yade adubu risk ama leire haidi moina karigumba khoigi ꯀꯩꯅꯣꯒꯨꯝꯕ ꯐꯦꯛꯇꯔ ꯏꯟꯚꯥꯏꯔꯣꯟꯃꯦꯟꯇꯦꯜ ꯐꯦꯛꯇꯔꯁꯤꯗ ꯁꯕꯁꯦꯞꯇꯦꯕꯜ ꯑꯣꯏꯔꯕ ꯃꯣꯏꯅ ꯉꯥꯛꯊꯣꯛꯄ ꯉꯝꯖ ꯇꯥꯔꯒꯗꯤ ꯖꯦꯅꯦꯇꯤꯛ ꯁꯤꯃꯤꯂꯔꯤꯇꯤ ꯂꯣꯏ ꯑꯣꯏꯔꯕꯅꯤꯅ ꯃꯣꯏꯁꯤ ꯃꯥꯡꯊꯔꯛꯄꯒꯤ ꯃꯣꯏꯅ ꯑꯦꯗꯖꯁ ꯇꯧꯕ ꯉꯝꯗꯕꯒꯤ ꯄꯣꯁꯤꯕꯤꯂꯤꯇꯤꯗꯤ ꯌꯥꯝ ꯋꯥꯡꯕ ꯍꯥꯏꯇꯥꯔꯦ ꯑꯗꯨꯅ ꯖꯦꯅꯦꯇꯤꯛ ꯗꯥꯏꯕꯔꯁꯤꯇꯤ ꯋꯥꯡꯉꯦ ꯀꯥꯟꯅꯕ ꯍꯥꯏꯇꯥꯔꯦ ꯑꯗꯨꯅ ꯑꯩꯈꯣꯏꯅ ꯁꯤꯒꯤ ꯑꯩꯒꯤ ꯔꯤꯁꯔꯁꯁꯤꯗ ꯑꯩꯈꯣꯏꯒꯤ ꯑꯣꯔꯀꯤꯗꯁꯇ ꯑꯩꯈꯣꯏ ꯌꯦꯡꯖꯔꯦ ꯃꯅꯤꯄꯨꯔꯒꯤꯀꯣ ꯑꯗꯒꯤ ꯕꯦꯝꯕꯨꯗ ꯌꯦꯡꯖꯔꯦ ꯑꯗꯣ ꯑꯣꯔꯀꯤꯗꯇ ꯑꯗꯨ ꯁꯤꯒꯤ ꯌꯥꯝꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯗꯤꯇꯦꯜꯁꯤꯕꯨꯗꯤ ꯑꯩꯒꯤ ꯂꯦꯕꯀꯤ ꯑꯩꯈꯣꯏꯒꯤ ꯔꯤꯁꯔꯁ ꯁ꯭ꯀꯣꯂꯔꯁꯤꯡꯅ ꯄꯤꯕꯤꯔꯅꯤꯀꯣ ꯑꯗ ꯑꯩꯈꯣꯏꯅ ꯍꯧꯖꯤꯛꯐꯥꯎ ꯐꯪꯕ ꯃꯇꯝꯁꯤꯗ ꯗꯥꯏꯕꯔꯁꯤꯇꯤꯁꯤ ꯌꯥꯝ ꯂꯣ ꯑꯣꯏ ꯍꯥꯏꯇꯥꯔꯦ ꯍꯧꯖꯤꯛꯁꯦ ꯑꯩꯈꯣꯏ ꯑꯣꯔꯀꯤꯗꯁ ꯏꯟꯗꯦꯟꯖꯔꯠꯅꯤ ꯀꯣ ꯑꯩꯈꯣꯏꯅ ꯈꯟꯒꯠꯆꯔꯤꯁꯦ ꯏꯟꯗꯦꯟꯖꯔ ꯁ꯭ꯄꯦꯁꯤꯁ ꯑꯣꯔꯀꯤꯗ ꯈꯟꯒꯠꯄꯅꯤ ꯌꯥꯝꯅ ꯂꯣ ꯑꯣꯏꯕꯅ ꯐꯪꯉꯤ ꯑꯗꯨꯅ ꯂꯣ ꯑꯣꯏꯕ ꯐꯪꯕꯁꯤꯅ ꯑꯗꯩ ꯑꯃꯨꯛꯁꯨ ꯑꯩꯈꯣꯏꯒꯤ ꯀꯩꯅꯣꯁꯦ ꯑꯩꯈꯣꯏꯒꯤ ꯃꯣꯏꯒꯤ ꯄꯣꯄꯨꯂꯦꯁꯟꯁꯤꯁꯨ ꯍꯟꯊꯔꯛꯄ ꯍꯥꯏꯗꯤ ꯗꯤꯐ꯭ꯔꯦꯟ ꯑꯩꯈꯣꯏꯒꯤ ꯐꯦꯛꯇꯔꯁ ꯃꯌꯥꯝ ꯂꯩꯔꯦ ꯑꯩꯈꯣꯏꯒꯤ ꯀ꯭ꯂꯥꯏꯃꯦꯠ ꯆꯦꯟꯖ ꯂꯥꯛꯄ ꯑꯗꯒꯤ ꯗꯤꯐꯣꯔꯦꯁꯇ꯭ꯔꯦꯁꯟ ꯑꯦꯛꯁꯄꯥꯟꯁꯟ ꯑꯣꯐ ꯁꯤꯇꯤꯁ ꯁꯤꯒꯨꯝꯕ ꯃꯌꯥꯝ ꯂꯩꯕꯁꯤꯅ ꯃꯈꯣꯏꯒꯤ ꯄꯣꯄꯨꯂꯦꯁꯟꯁꯨ ꯍꯟꯊꯔꯛꯄ ꯄꯣꯄꯨꯂꯦꯁꯟ ꯍꯟꯊꯕꯇ ꯅꯠꯇꯅ ꯑꯃꯨꯛꯁꯨ ꯃꯥꯒꯤ ꯉꯁꯥꯏꯒꯤ ꯗꯥꯏꯕꯔꯁꯤꯇꯤ ꯂꯣ ꯑꯣꯏꯔꯛꯄꯁꯤꯗꯤ ꯗꯤꯁ ꯏꯁ ꯚꯦꯔꯤ ꯚꯦꯔꯤ ꯗꯦꯟꯖꯔꯁ ꯑꯗꯒꯤ ꯁꯤꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯗꯥꯏꯕꯔꯁꯤꯇꯤꯒꯤ ꯑꯣꯏꯔꯦ ꯑꯗꯒꯤ ꯑꯩꯈꯣꯏꯅ ꯍꯧꯖꯤꯛ ꯍꯧꯖꯤꯛ ꯑꯩꯈꯣꯏꯅ ꯑꯃꯨꯛ ꯁꯥꯏꯠ ꯑꯃꯗ ꯑꯩꯈꯣꯏꯅ ꯄꯥꯏꯈꯠꯆꯔꯤꯁꯨ ꯁꯤꯁꯨ ꯑꯣꯜꯃꯣꯁ ꯀꯟꯖꯥꯔꯕꯦꯁꯟꯒ ꯔꯤꯂꯦꯇꯦഡ് ꯑꯣꯏꯕꯅꯤ ꯑꯁꯤꯗ ꯑꯩꯈꯣꯏꯅ ꯏꯟ ꯕꯤꯇ꯭ꯔꯣ ꯄ꯭ꯔꯣꯄꯒꯦꯁꯟ ꯄ꯭ꯂꯥꯟ ꯇꯤꯁꯨ ꯀꯜꯆꯔ ꯇꯧꯔꯒ ꯍꯥꯏꯗꯤ ꯀꯩꯅꯣꯗ ꯉꯁꯥꯏꯒꯤ ꯏꯟꯗꯦꯟꯖꯔ ꯑꯣꯏꯔꯤꯕ ꯍꯧꯖꯤꯛ ꯐꯪꯖꯜꯂꯛꯇ꯭ꯔꯤꯕ ꯄ꯭ꯂꯥꯟ ꯃꯌꯥꯝꯁꯤ ꯑꯩꯈꯣꯏꯅ ꯌꯣꯛꯈꯠꯅꯕ ꯃꯁꯤꯡ ꯍꯦꯟꯒꯠꯅꯕ ꯁꯤꯁꯨ ꯑꯩꯈꯣꯏꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯀꯩꯅꯣ ꯇꯧꯕ ꯗꯔꯀꯥꯔ ꯑꯣꯏꯌꯦꯕ ꯃꯁꯤ ꯑꯩꯈꯣꯏꯅ ꯏꯟ ꯕꯤꯇ꯭ꯔꯣ ꯇꯤꯁꯨ ꯀꯜꯆꯔ ꯇꯦꯛꯅꯤꯛ ꯁꯤꯖꯤꯟꯅꯔꯒ ꯑꯩꯈꯣꯏꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯏꯟ ꯕꯤꯇ꯭ꯔꯣ ꯄ꯭ꯔꯣꯇꯣꯀꯣꯜ ꯍꯥꯏꯇꯥꯔꯦ ꯍꯥꯏꯗꯤ ꯃꯣꯏꯁꯦ ꯀꯃꯥꯏꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯂꯦꯕꯇ ꯀꯃꯥꯏꯅ ꯃꯣꯏꯁꯦ ꯍꯧꯍꯟꯒꯗꯒꯦ ꯏꯟ ꯕꯤꯇ꯭ꯔꯣ ꯀꯟꯗꯤꯁꯟꯗ ꯑꯗꯨꯃꯥꯏꯅ ꯇꯧꯗꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯄ꯭ꯔꯣꯇꯣꯀꯣꯜ ꯃꯌꯥꯝ ꯑꯃ ꯏꯁꯇꯥꯕ꯭ꯂꯤꯁ ꯇꯧꯖꯈ꯭ꯔꯦ ꯑꯗꯨꯒꯤ ꯐꯥꯏꯟꯗꯤꯡꯗꯨꯁꯨ ꯑꯩꯈꯣꯏꯅ ꯀꯩꯅꯣꯗ ꯑꯩꯈꯣꯏꯒꯤ ꯏꯟꯇꯔꯅꯦꯁꯅꯦꯜ ꯖꯔꯅꯦꯜꯁꯤꯡꯗ ꯑꯩꯈꯣꯏ ꯄꯕ꯭ꯂꯤꯁ ꯇꯧꯖꯔꯦ
ꯑꯩꯒꯤ ꯃꯤꯡꯅ ꯗꯣꯛꯇꯔ ꯂꯦꯡꯕꯝ ꯇꯤꯀꯦꯟꯗ꯭ꯔ ꯁꯤꯡ ꯗꯤꯄꯥꯔꯠꯃꯦꯟ ꯑꯣꯐ ꯕꯥꯏꯑꯣꯇꯦꯛꯅꯣꯂꯣꯖꯤ ꯃꯅꯤꯄꯨꯔ ꯌꯨꯅꯤꯚꯔꯁꯤꯇꯤꯗ ꯒꯦꯁ ꯐꯦꯀꯜꯇꯤ ꯑꯣꯏꯅ ꯂꯩꯖꯕꯅꯤ ꯁꯤꯒꯤ ꯑꯩꯈꯣꯏ ꯄ꯭ꯂꯥꯟ ꯕꯥꯏꯑꯣꯇꯦꯛꯅꯣꯂꯣꯖꯤꯒꯤ ꯂꯦꯕꯣꯔꯦꯇꯔꯤꯁꯤꯗ ꯑꯩꯈꯣꯏꯅ ꯇꯧꯔꯤꯕ ꯋꯥꯛꯁꯤ ꯁꯤꯗ ꯃꯣꯂꯤꯀꯨꯂꯔ ꯋꯥꯛꯅꯤ ꯃꯣꯂꯤꯀꯨꯂꯔ ꯋꯥꯛꯁꯤ ꯑꯩꯈꯣꯏ ꯄꯥꯝꯕꯤꯁꯤꯡꯒꯤ ꯃꯈꯣꯏꯒꯤ ꯄꯥꯔꯠ ꯃꯔꯨꯑꯣꯏꯅ ꯃꯅꯥꯗꯒꯤ ꯑꯩꯈꯣꯏꯒꯤ DNA ꯑꯦꯛꯁꯇ꯭ꯔꯦꯛꯁꯟ ꯇꯧꯔꯀꯥꯟꯗ ꯑꯦꯛꯁꯇ꯭ꯔꯦꯛ ꯇꯧꯔꯤꯕ DNA ꯑꯗꯨ ꯑꯩꯈꯣꯏ ꯃꯥꯒꯤ ꯀ꯭ꯋꯥꯂꯤꯇꯤ ꯇꯦꯁ ꯇꯧꯅꯕꯒꯤ ꯑꯁꯤꯒꯤ ꯖꯦꯜ ꯏꯂꯦꯛꯇ꯭ꯔꯣꯐꯣꯔꯦꯁ ꯁꯤꯖꯤꯟꯅꯔꯒ ꯑꯩꯈꯣꯏ ꯖꯦꯜ ꯃꯥꯒꯤ ꯀ꯭ꯋꯥꯂꯤꯇꯤ ꯇꯦꯁ ꯇꯧꯔꯦ ꯑꯗꯣ ꯑꯩꯈꯣꯏꯒꯤ ꯑꯗꯨꯒꯤ ꯃꯈꯥ ꯇꯥꯕ ꯑꯩꯈꯣꯏꯒꯤ ꯗꯥꯎꯟꯁ꯭ꯇ꯭ꯔꯤꯝ ꯄ꯭ꯔꯣꯁꯦꯁꯤꯡ ꯑꯣꯏꯅ ꯃꯈꯥ ꯇꯥꯕ ꯁ꯭ꯇꯦꯞꯁꯤꯡꯗ ꯑꯩꯈꯣꯏꯅ ꯍꯥꯏꯔꯤꯕ ꯖꯤꯅꯣꯃꯤꯛ DNA ꯁꯤꯖꯤꯟꯅꯔꯀꯥꯟꯗ PCR ꯃꯦꯆꯤꯟ ꯑꯁꯤꯗꯒꯤ ꯃꯇꯦꯡꯅ ꯑꯩꯈꯣꯏꯒꯤ DNAꯒꯤ ꯕꯦꯟꯁꯤꯡꯁꯤ ꯑꯦꯝꯄ꯭ꯂꯤꯐꯥꯏ ꯇꯧꯔꯦ ꯑꯗꯨꯒ ꯑꯩꯈꯣꯏꯅ ꯈꯪꯂꯤꯕ ꯁꯦꯝꯄꯜꯁꯤꯡ ꯃꯌꯥꯝꯁꯤꯡꯁꯤꯒꯤ ꯃꯔꯛꯇ ꯂꯩꯔꯤꯕ ꯑꯩꯈꯣꯏ ꯈꯦꯠꯅꯕ ꯀꯌꯥꯝ ꯂꯩ ꯑꯗꯨꯒ ꯇꯤꯁꯨ ꯀꯜꯆꯔ ꯇꯧꯕ ꯄꯥꯝꯕꯤ ꯑꯣꯏꯇꯥꯔꯒꯗꯤ ꯃꯈꯣꯏꯒꯤ ꯃꯔꯛꯇ ꯁꯨꯃꯥꯀ꯭ꯂꯣꯅꯦꯜ ꯚꯦꯔꯤꯌꯦꯁꯟ ꯀꯌꯥꯝ ꯊꯣꯛꯏ ꯊꯣꯛꯇꯦ ꯍꯥꯏꯕꯁꯤꯡ ꯃꯈꯣꯏꯒꯤ ꯄꯣꯂꯤꯃꯣꯔꯐꯤꯖꯝ ꯃꯣꯅꯣꯃꯣꯔꯐꯤꯖꯝ ꯆꯦꯛ ꯇꯧꯏ ꯑꯗꯨ ꯄꯣꯄꯨꯂꯦꯁꯟꯒꯤ ꯑꯣꯏꯅ ꯈꯣꯝꯃꯛꯄ ꯄꯣꯄꯨꯂꯦꯁꯟ ꯇꯣꯉꯥꯟ ꯇꯣꯉꯥꯟꯕ ꯄꯣꯄꯨꯂꯦꯁꯟ ꯈꯣꯝꯃꯛꯄꯁꯤꯡꯒꯤ ꯃꯔꯛꯇ ꯁ꯭ꯄꯦꯁꯤꯁꯁꯤꯡꯁꯤꯒꯤ ꯀꯔꯤ ꯈꯦꯠꯅꯕ ꯂꯩꯒꯦ ꯍꯥꯏꯕꯗꯨ ꯑꯩꯈꯣꯏ PCRꯗꯒꯤ ꯑꯦꯝꯄ꯭ꯂꯤꯐꯥꯏ ꯇꯧꯔꯕ ꯕꯦꯟꯁꯤꯡꯗꯨ ꯖꯦꯜ ꯗꯣꯀꯨꯃꯦꯟꯇꯦꯁꯟ ꯁꯤꯁꯇꯦꯝꯁꯤꯗꯒꯤ ꯑꯩꯈꯣꯏ ꯃꯥꯒꯤ ꯖꯦꯜ ꯏꯃꯦꯖꯁꯤ ꯏꯂꯦꯛꯇ꯭ꯔꯣꯐꯣꯔꯦꯁꯤꯁꯅ ꯔꯟ ꯇꯧꯕ ꯂꯣꯏꯔꯕ ꯖꯦꯜ ꯏꯃꯦꯖ ꯑꯁꯤꯗꯒꯤ ꯑꯩꯈꯣꯏꯅ ꯁ꯭ꯇꯗꯤ ꯇꯧꯕꯅꯤ
ꯑꯩꯈꯣꯏꯅ ꯎꯅꯖꯃꯤꯁꯛ ꯑꯁꯤ ꯑꯩꯈꯣꯏ ꯗꯤꯄꯥꯔꯠꯃꯦꯟ ꯑꯁꯤꯗ ꯊꯕꯛ ꯇꯧꯕꯤꯔꯤꯕ ꯑꯣꯖꯥꯁꯨ ꯑꯣꯏꯕꯤꯔꯤꯕ ꯑꯩꯈꯣꯏꯒꯤ ꯗꯣꯛꯇꯔ ꯍꯃꯤꯗꯨꯔ ꯔꯍꯃꯥꯟꯒꯅꯤ ꯑꯦꯁꯤꯁꯇꯦꯟꯠ ꯄ꯭ꯔꯣꯐꯦꯁꯔ ꯑꯍꯥꯟꯕꯗ ꯑꯣꯖꯥꯕꯨ ꯗꯨꯔꯗꯔꯁꯟ ꯃꯅꯤꯄꯨꯔꯤ ꯃꯥꯏꯀꯩꯗꯒꯤ ꯇꯔꯥꯝꯅ ꯑꯣꯛꯆꯔꯤ ꯑꯍꯥꯟꯕꯁꯤꯗꯤ ꯑꯣꯖꯥ ꯍꯧꯖꯤꯛ ꯏꯁꯥꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯔꯤꯁꯔꯁ ꯇꯧꯔꯤꯕ ꯑꯦꯔꯤꯌꯥꯁꯤ ꯀꯔꯝ ꯑꯦꯔꯤꯌꯥꯗꯣ ꯁꯥꯔꯅ ꯇꯧꯕꯤꯔꯤꯁꯦ
ꯑꯩꯈꯣꯏꯅ ꯃꯔꯨꯑꯣꯏꯅ ꯇꯧꯖꯔꯤꯁꯤ structure and function studies of protein such as antioxidant enzyme asidani maruoina aikoina tawjarisi maruoina protein si any genes ki oiba aroiba product si protein ni aduna protein haisina magi varied function mayam am lai maruoina yamna samna haijarabada aikoina maruoina tawjarisi protein antioxidant enzyme aikoina maruoina focus tawjarisi masi disease kya marumde involved toba adugumba enzymes amang khaktani aikoigi any pathogen asina haksangda changaba matungda pet haksangi oba defense mechanism mayamama aikoi molecules mayamama samna harabada aikoigi highly reactive oxygen species mayamam thadore aikoigi mahiksi pathogen si hatoknaba adu matamda maruoina aikoigi leiriba disease tugi organism haitare mycobacterium haire natragana malaria gida hargana parasite tuna mana ꯑꯩꯈꯣꯏꯒꯤ ꯗꯤꯐꯦꯟꯁ ꯃꯦꯀꯥꯅꯤꯖꯝ ꯑꯃ ꯄꯨꯊꯣꯔꯛꯑꯦꯕ ꯍꯥꯏꯂꯤ ꯔꯤꯑꯦꯛꯇꯤቭ ꯑꯣꯛꯁꯤꯖꯟ ꯁ꯭ꯄꯦꯁꯤꯁ ꯍꯛꯆꯥꯡꯅ ꯄꯤꯊꯣꯔꯛꯏꯗꯣ ꯑꯗꯨ ꯗꯤꯐꯦꯟꯁ ꯇꯧꯅꯕ ꯑꯗꯨꯗ ꯑꯦꯟꯇꯤ ꯑꯣꯛꯁꯤꯗꯦꯟ ꯏꯟꯖꯥꯏꯝꯁꯤ ꯃꯔꯨꯑꯣꯏꯅ ꯃꯗꯨꯗ ꯌꯥꯎꯕ ꯏꯟꯖꯥꯏꯝ ꯑꯃꯈꯛꯅꯦꯕ ꯑꯗꯣ ꯑꯩꯈꯣꯏꯅ ꯃꯔꯨꯑꯣꯏꯅ ꯐꯣꯀꯁ ꯇꯧꯖꯔꯤꯗꯤ ꯄꯥꯔ ꯑꯣꯛꯁꯤ ꯔꯦꯗꯣꯛꯁꯤꯟ 6ꯅꯤ ꯃꯁꯤ ꯃꯦꯂꯦꯔꯤꯌꯥꯗꯁꯨ ꯃꯁꯤ ꯈꯛꯇꯅꯤ ꯑꯌꯥꯝꯕꯅ ꯑꯦꯟꯇꯤ ꯑꯣꯛꯁꯤꯗꯦꯟ ꯏꯟꯖꯥꯏꯝ ꯑꯣꯏꯅ ꯌꯥꯎꯖꯔꯤꯁꯦ ꯑꯗꯨꯅ ꯑꯩꯈꯣꯏꯅ ꯃꯁꯤ ꯍꯧꯖꯤꯛ ꯑꯩꯈꯣꯏꯒꯤ ꯑꯩꯒꯤ ꯁꯥꯇ꯭ꯔꯁꯤꯡꯅ ꯃꯈꯣꯏꯅ ꯇꯥꯛꯄꯤꯒꯅꯤ ꯃꯔꯝꯗꯤ ꯃꯈꯣꯏꯅ ꯍꯧꯖꯤꯛ ꯄ꯭ꯔꯣꯇꯤꯟꯁꯦ ꯑꯩꯈꯣꯏ ꯀ꯭ꯂꯣꯟ ꯇꯧꯔꯒ ꯕꯦꯛꯇꯔꯤꯌꯦꯜ ꯁꯤꯁꯇꯦꯝꯗ ꯑꯩꯈꯣꯏ ꯍꯧꯖꯤꯛ ꯑꯦꯛꯁꯄ꯭ꯔꯦꯁ ꯇꯧꯔꯦ ꯑꯩꯈꯣꯏꯒꯤ ꯄꯥꯟꯗꯝꯗꯤ ꯀꯩꯅꯣꯝꯇ ꯑꯦꯛꯁꯄ꯭ꯔꯦꯁ ꯇꯧꯔ ꯃꯇꯨꯡꯗ ꯃꯥꯒꯤ ꯁ꯭ꯇ꯭ꯔꯛꯆꯔꯁꯤ ꯁ꯭ꯇꯗꯤ ꯇꯧꯕ ꯑꯗꯨꯒ ꯃꯉꯣꯟꯗ ꯇꯥꯔꯒꯦꯠ ꯇꯧꯕ ꯌꯥꯕ ꯁꯥꯏꯠꯁꯦ ꯊꯤꯕ ꯑꯗꯨꯅ ꯑꯩꯈꯣꯏꯅ ꯃꯔꯨꯑꯣꯏꯅ ꯇꯧꯗꯣꯔꯁꯤ ꯁꯥꯏꯠꯇ ꯊꯤꯔꯒ ꯑꯩꯈꯣꯏꯅ ꯗ꯭ꯔꯒ ꯁꯤꯟꯊꯦꯁꯤꯁ ꯇꯧꯔꯒ ꯃꯗꯨ ꯗ꯭ꯔꯒꯇꯨ ꯑꯩꯈꯣꯏ ꯄ꯭ꯂꯥꯟ ꯁꯣꯔꯁꯦꯁ ꯑꯣꯏꯕ ꯌꯥꯏ ꯅꯠꯇ꯭ꯔꯒꯅ ꯑꯦꯅꯤ ꯑꯇꯣꯞꯄ ꯃꯥꯏꯀ꯭ꯔꯣꯕꯦꯛꯇꯔꯤꯌꯝ ꯁꯣꯔꯁꯦꯁ ꯑꯣꯏꯕ ꯌꯥꯏ ꯑꯩꯈꯣꯏꯅ ꯃꯗꯨ ꯁꯤꯟꯊꯦꯁꯥꯏꯖ ꯇꯧꯕ ꯗꯦꯔꯤꯚꯦꯇꯥꯏꯖ ꯇꯧꯕ ꯑꯗꯨꯗꯨꯒ ꯃꯉꯣꯟꯗ ꯑꯩꯈꯣꯏ ꯇꯥꯔꯒꯦꯠ ꯇꯧꯔꯒ ꯑꯩꯈꯣꯏꯒꯤ ꯉꯁꯥꯏꯒꯤ ꯑꯦꯟꯇꯤ ꯑꯣꯛꯁꯤꯗꯦꯟ ꯏꯟꯖꯥꯏꯝꯁꯤ ꯀꯔꯤꯒꯨꯝꯕ ꯑꯩꯈꯣꯏꯅ ꯇꯥꯔꯒꯦꯠ ꯇꯧꯕ ꯉꯝꯃꯒꯗꯤ ꯄ꯭ꯔꯣꯒ꯭रेꯁꯟ ꯑꯣꯐ ꯗꯤ ꯗꯤꯖꯤꯖꯁꯦ ꯑꯩꯈꯣꯏꯅ ꯕ꯭ꯂꯣꯛ ꯇꯧꯕ ꯉꯝꯒꯅꯦꯕ ꯄ꯭ꯔꯣꯒ꯭रेꯁꯟ ꯑꯣꯐ ꯗꯤꯖꯤꯖꯁꯦ ꯑꯩꯈꯣꯏ ꯃꯈꯟ ꯃꯈꯟꯗ ꯁ꯭ꯇꯦꯖ ꯇꯣꯉꯥꯟꯕꯗ ꯕ꯭ꯂꯣꯛ ꯇꯧꯕ ꯌꯥꯏ ꯍꯥꯏꯕꯗꯤ ꯖꯤꯅꯣꯝ ꯂꯦꯚꯦꯜꯗ ꯇꯧꯕ ꯌꯥꯏ ꯅꯠꯇ꯭ꯔꯒ ꯄ꯭ꯔꯣꯇꯤꯌꯣꯝ ꯂꯦꯚꯦꯜꯗ ꯇꯧꯕ ꯌꯥꯏ ꯑꯩꯈꯣꯏꯅ ꯇꯧꯖꯔꯤꯁꯤꯗꯤ ꯄ꯭ꯔꯣꯇꯤꯌꯣꯃꯤꯛ ꯂꯦꯚꯦꯜꯗꯅꯦꯕ ꯍꯧꯖꯤꯛ ꯇꯧꯔꯤꯁꯦ ꯃꯁꯤ ꯍꯥꯏꯕꯗꯤ ꯃꯁꯤꯅ ꯌꯥꯝꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯄ꯭ꯔꯣꯒ꯭रेꯁꯟ ꯑꯣꯐ ꯗꯤ ꯗꯤꯖꯤꯖꯁꯤꯗ ꯕ꯭ꯂꯣꯛ ꯇꯧꯅꯅꯕꯒꯤ ꯌꯥꯝꯅ ꯑꯆꯧꯕ ꯊꯤꯡꯅꯅꯕꯒꯤ ꯑꯆꯧꯕ ꯊꯧꯗꯥꯡ ꯑꯃ ꯑꯣꯏꯔꯛꯄꯁꯨ ꯌꯥꯏ ꯑꯗꯨꯃꯛꯅ ꯑꯩꯈꯣꯏꯅ ꯍꯧꯖꯤꯛ ꯇꯧꯖꯔꯤꯁꯤꯗꯤ ꯑꯗꯨꯒ ꯍꯧꯖꯤꯛꯃꯛ ꯑꯇꯣꯞꯄ ꯑꯃꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯃꯤꯁꯐꯣꯜꯗꯤꯡ ꯑꯣꯐ ꯗꯤ ꯗꯤꯖꯤꯖ ꯃꯤꯁꯐꯣꯜꯗꯤꯡ ꯗꯤꯖꯤꯖ ꯍꯥꯏꯕꯗꯤ ꯅ꯭ꯌꯨꯔꯣꯗꯤꯖꯦꯅꯦꯔꯦꯇꯤቭ ꯗꯤꯖꯤꯖ ꯑꯜꯖꯥꯏꯃꯔꯒꯨꯝꯕ ꯄꯥꯔꯀꯤꯟꯁꯟꯒꯨꯝꯕ ꯃꯁꯤ ꯏꯟꯗꯤꯌꯥꯗꯁꯨ ꯃꯤ ꯂꯥꯛ ꯑꯃꯗ ꯃꯤ 4,000,000ꯃꯨꯛꯅꯦꯕ ꯅꯥꯔꯤꯁꯦ ꯂꯥꯛ ꯑꯃꯃꯝꯗ ꯑꯗꯨꯗꯤ ꯁꯝꯅ ꯍꯥꯏꯔꯕꯗ 2050ꯗ ꯑꯣꯜ ꯑꯣꯚꯔ ꯋꯥꯔꯜꯒꯤ 150 ꯃꯤꯂꯤꯌꯟꯒꯨꯝꯕ ꯅꯥꯅꯦꯅ ꯑꯩꯈꯣꯏꯅ ꯄ꯭ꯔꯤꯗꯤꯛ ꯇꯧꯔꯤꯕ ꯑꯦꯛꯁꯄꯦꯛ ꯇꯧꯔꯤꯕ ꯁꯤ ꯀꯔꯝꯃꯥꯏꯅ ꯇꯧꯏ ꯍꯥꯏꯕ ꯃꯇꯝꯗ ꯑꯩꯈꯣꯏꯒꯤ ꯄ꯭ꯔꯣꯇꯤꯟ ꯃꯌꯥꯝ ꯑꯃꯁꯤ ꯃꯤꯁꯐꯣꯜ ꯇꯧꯕ ꯑꯍꯟ ꯑꯣꯏꯔꯛꯄ ꯃꯇꯝꯗ ꯃꯥꯒꯤ ꯐꯪꯁꯟꯁꯤ ꯃꯔꯨꯑꯣꯏꯅ ꯄ꯭ꯔꯣꯇꯤꯟꯁꯤ ꯐꯪꯁꯟ ꯇꯧꯗ꯭ꯔꯒꯗꯤ ꯃꯥꯁꯤ ꯃꯤꯁꯐꯣꯜ ꯇꯧꯔꯛꯏ ꯑꯗꯨꯗꯨꯒ ꯃꯗꯨꯅ ꯗꯤꯖꯤꯖ ꯑꯃ ꯑꯣꯏꯍꯜꯂꯦꯕ ꯑꯗꯨꯅ ꯃꯔꯨꯑꯣꯏꯅ ꯑꯩꯈꯣꯏ ꯏꯄꯤꯂꯦꯞꯁꯤꯒꯨꯝꯕ ꯑꯗꯒꯤ ꯀꯥꯎꯒꯟꯕꯒꯨꯝꯕ ꯑꯩꯈꯣꯏ ꯑꯍꯟꯁꯤꯡꯗ ꯇꯧꯔꯤꯁꯦ ꯑꯍꯟ ꯑꯣꯏꯔꯛꯄ ꯃꯇꯝꯗ ꯁꯤꯒꯨꯝꯕ ꯃꯌꯥꯝꯁꯤ ꯊꯣꯛꯍꯜꯂꯦꯕ ꯁꯤ ꯍꯧꯖꯤꯛ ꯑꯩꯈꯣꯏꯅ ꯀꯃꯥꯏꯅ ꯇꯧꯏ ꯍꯥꯏꯕ ꯃꯇꯝꯗ ꯃꯁꯤ ꯄ꯭ꯂꯥꯟꯒꯤ ꯁꯣꯔꯁꯇꯒꯤ ꯂꯧꯔꯒ ꯁꯤꯒꯤ ꯃꯤꯁꯐꯣꯜꯁꯤ ꯀꯔꯦꯛ ꯇꯧꯅꯅꯕꯒꯤ ꯑꯩꯈꯣꯏꯒꯤ ꯃꯣꯂꯤꯀꯨꯜꯁꯤꯡ ꯑꯩꯈꯣꯏ ꯍꯧꯖꯤꯛ ꯑꯩꯈꯣꯏꯒꯤ ꯃꯔꯨꯑꯣꯏꯅ ꯑꯩꯈꯣꯏ ꯃꯗꯨꯃ ꯑꯩꯈꯣꯏꯒꯤ ꯂꯧꯈꯠꯆꯔꯤ ꯑꯩꯈꯣꯏꯒꯤ ꯂꯦꯕꯁꯤꯗ ꯍꯥꯏꯕꯗꯤ ꯄ꯭ꯂꯥꯟꯁꯤꯡꯁꯦ ꯑꯦꯛꯁꯇ꯭ꯔꯦꯛ ꯇꯧꯔꯒ ꯇꯣꯉꯥꯟ ꯇꯣꯉꯥꯟꯕ ꯂꯦꯚꯦꯜꯗ ꯑꯩꯈꯣꯏꯅ ꯄ꯭ꯌꯨꯔꯤꯐꯥꯏ ꯇꯧꯔꯒ ꯑꯗꯨꯒꯨꯝꯕ ꯃꯣꯂꯤꯀꯨꯜꯁꯤꯡꯗꯨ ꯑꯩꯈꯣꯏꯒꯤ ꯄ꯭ꯌꯨꯔꯤꯐꯥꯏ ꯇꯧꯕ ꯍꯥꯏꯕꯗꯤ GCMS ꯑꯗꯒꯤ HPLC ꯁꯤꯒꯨꯝꯕ ꯃꯌꯥꯝꯁꯤ ꯁꯤꯖꯤꯟꯅꯔꯒ ꯀꯦꯔꯦꯛꯇꯔꯥꯏꯖ ꯇꯧꯔꯒ ꯇꯥꯔꯒꯦꯠ ꯇꯧꯕ ꯑꯩꯈꯣꯏꯒꯤ ꯃꯤꯁꯐꯣꯜ ꯇꯧꯔꯤꯕ ꯄ꯭ꯔꯣꯇꯤꯟꯁꯤꯡꯗꯣ ꯑꯗꯨꯒ ꯃꯈꯣꯏꯕꯨ ꯔꯦꯛꯇꯤꯐꯥꯏ ꯇꯧꯔꯒ ꯀꯔꯦꯛ ꯐꯣꯜꯗꯤꯡꯗ ꯂꯥꯛꯍꯜꯂꯒ ꯗꯤꯖꯤꯖꯇꯣ ꯑꯩꯈꯣꯏꯅ ꯀꯟꯇ꯭ꯔꯣꯜ ꯇꯧꯅꯕ ꯍꯣꯠꯅꯕ ꯑꯗꯨꯁꯨ ꯑꯇꯣꯞꯄ ꯑꯩꯈꯣꯏꯒꯤ ꯂꯦꯕꯁꯤꯗ ꯇꯧꯕ ꯀꯔꯤ ꯔꯤꯁꯔꯁ ꯑꯃꯅꯤ
ꯁꯔ ꯑꯩꯈꯣꯏꯒꯤ ꯍꯧꯖꯤꯛꯀꯤ ꯀꯣꯚꯤꯗ ꯃꯇꯥꯡ ꯅꯩꯅꯔꯛꯄꯁꯦ ꯃꯁꯤ ꯑꯦꯅꯥꯂꯥꯏꯁꯤꯁ ꯇꯧꯕꯁꯤꯁꯨ ꯃꯇꯦꯡ ꯑꯗꯨꯝ ꯑꯣꯏꯕ꯭ꯔꯥ ꯁꯔ
ꯃꯁꯤꯁꯨ ꯑꯣꯏꯒꯅꯤ ꯃꯔꯝꯗꯤ ꯀꯩꯅꯣꯗ COVID-19 ꯑꯁꯤ ꯚꯥꯏꯔꯦꯜ ꯗꯤꯖꯤꯖ ꯑꯃꯅꯤ ꯃꯥꯁꯤ ꯀꯟꯇ꯭ꯔꯣल ꯇꯧꯒꯦ ꯍꯥꯏꯔꯒꯗꯤ ꯃꯉꯣꯟꯗ ꯌꯥꯎꯔꯤꯕ ꯖꯤꯅꯣꯝ ꯂꯦꯚꯦꯜ ꯅꯠꯇ꯭ꯔꯒ ꯄ꯭ꯔꯣꯇꯤꯌꯣꯝ ꯂꯦꯚꯦꯜꯗ ꯑꯩꯈꯣꯏꯅ ꯇꯥꯔꯒꯦꯠ ꯇꯧꯕ ꯇꯥꯏꯕ ꯑꯩꯈꯣꯏꯒꯤ ꯁ꯭ꯄꯥꯏꯛ ꯑꯩꯅ ꯍꯧꯖꯤꯛ ꯌꯥꯝ ꯀꯟꯅ ꯃꯔꯨꯑꯣꯏꯅ ꯑꯩꯈꯣꯏ COVID-19ꯗ ꯇꯥꯔꯒꯦꯠ ꯑꯣꯏꯔꯤꯕ ꯃꯐꯝꯁꯤ ꯄ꯭ꯔꯣꯇꯤꯟꯅꯤ ꯃꯥꯁꯤ ꯀꯩꯒꯨꯝꯕ ꯑꯩꯈꯣꯏꯅ ꯁ꯭ꯇ꯭ꯔꯛꯆꯔꯁꯤ ꯁ꯭ꯇꯗꯤ ꯇꯧꯕ ꯉꯝꯗ꯭ꯔꯒꯗꯤ ꯑꯩꯈꯣꯏꯅ ꯀꯔꯝꯍꯥꯏꯅ ꯇꯥꯔꯒꯦꯠ ꯇꯧꯒꯅꯤ ꯑꯗꯨꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯂꯦꯕꯇ ꯃꯔꯨꯑꯣꯏꯅ ꯇꯥꯔꯒꯦꯠ ꯃꯔꯨꯑꯣꯏꯅ ꯁ꯭ꯇꯗꯤ ꯇꯧꯔꯤꯁꯤ ꯑꯇꯩ ꯅꯠꯇꯦ ꯉꯁꯥꯏꯒꯤ ꯁ꯭ꯇ꯭ꯔꯛꯆꯔꯁꯤ तोंगा तोंगा मेथड सिंग मरुइना बायोफिजिकल मेथड सिंग मरुइना एक्सरे क्रिस्टोलोग्राफी सुगुम्बा सिजनरगा अखोई कैरेक्टराइज तौनबा होनबा अदुसु कोविड-19 युसु अखोई मसिदी अखोई मणिपुरदरी खुदोंग चाबदु याम थोइना लेतबनीना अखोइना अतोपा लेब सिंगा कम्पीट वर्ल्ड गे अतोपा लेब सिंगा कम्पीट तौबा ngamdabdagi अखोइना मदुमक पाइखटपा ngamjadragasu अखोइसु मागी स्ट्रक्चर सि स्टडी तौनबा कन्ना अखोइसु होनरी समना हाबदे अखोइगी डॉक्टर हेरोजित सिंह मासी NII दगी अगी अखोइसिदा RA ओइना थबक तौबने मरोइना मदुदे स्पेशलाइज तौबा रिसर्च स्कॉलर अमनी मासी रिसर्च एसोसिएट अमनी मागी मतेंगना तो अखोइ लैरिबा रिसर्च स्कॉलर सिंगसि क्लोनिंग दगी हौरगा अखोइ प्रोटीन क्रिस्टलाइज फोबा अखोइगी लैब सिदा एक्सपेरिमेंट रिसर्च तौननबा कन्ना अखोइ कन होनरी अदुगा तौसु तौरी
ꯑꯩꯒꯤ ꯃꯤꯡ ꯗꯣꯛꯇꯔ ꯍꯦꯔꯣꯖꯤꯠ ꯑꯩ ꯂꯦꯕ ꯑꯁꯤꯗ ꯅꯦꯁꯅꯦꯜ ꯄꯣꯁꯠ ꯗꯣꯛꯇꯣꯔꯦꯜ ꯐꯦꯂꯣꯁꯤꯞ ꯐꯪꯖꯔꯒ ꯂꯩꯕ ꯑꯩꯈꯣꯏꯒꯤ ꯄꯣꯁꯠ ꯗꯣꯛꯇꯣꯔꯦꯜꯅꯤ ꯍꯥꯏꯇꯥꯔꯦ दुगा अखोइना मरोइना लैब सिदा तौजरिबसे अखोइगे स्ट्रक्चर अमदी फंक्शनल बायोलॉजी लैब नि मसिदा अखोइना प्रोटीन गी अखोइना फंगकपा अखोइना वर्क तौरिबा मरोइना एंटीऑक्सीडेंट प्रोटीन खरगी स्ट्रक्चर डिटरमाइन तौरगा मदुना कमदौना मदुदा करी करी मॉडिफिकेशन थोकलगा अखोइना मदुदा हयेंग कोना सारकपा याबा इनहिबिटर्स अमदी ड्रग ना सिंगबा साबगी मरमदा अखोइ वर्क तौजे अखोइगे मरोइना प्रोजेक्टसिंग असिदा Department of Biotechnology Government of India amadi DST Department of Science and Technology की funding की मखा दे कोई तौजबनी पोस्ट डॉक्टोरल की अमदी अतई प्रोजेक्ट खरा अमदी मसीदा मखोइना Government of India कोई ना थागत पसु फोंदोकजरी
Department sida koine assistant professor nu pubire bi khogi Dr. Rina Hawam gani ahan bada Doordarshan Manipur taram noksir dam
thak chai
ahan bas madam huzik karam area du si madam huzik work to biri se
hausi khoigi lab sida project mari gumba tawjari ada project se koina ahum amadi gene polymorphism hai tare koigi population amada gene amaga mi amaga individually gene khena base siga koigi disease kharaga kamaina mari leina ge hapadu sum thijan ba hai tare hamba project ta coin to ise gene khara drug addiction ga mari leina ba gene khara da koi gi drug addict singa addict oida ba mi singa karamba khena ba lai ba ge hapadu thijan jakhre masi DBT na sponsor to bar project ni tuning program gi makhada aduga masi da koina major oina phangja ge badi gene ama koina target tawjaba dopamine transporter haba gene sida sequence kara khena badagi koi gi drug addict population sing se sidana hen risk factor laiba genotype kara koina thengna je control singa compare tawba yengba matamda adagi mathang da koina TB gi naba TB naba patient singda koina moigi hepatotoxicity na liver narakpa moigi drug yam strong oba drug sing se sar kanda narakpa hepatotoxicity siga koigi drug metabolize to boda related oba gene kharaga kamaina mari leina ge hepato check tawjare masida koigi population sida hoji publicity tawri results se koina phangjare aduda koigi population sida koigi GST na gene amale glutathione S transferase na masida koina genotype khara risk factor laiba genotype khara su observe tawjare adu hojik data analyze or koi publish tawna ba tawjari adagi amaromda koina traditionally seasonaba plant sing se medicinal plant sing sidagi koi singli nauri ki lying bada seasonaba plant khara koina crude extract oina isolate tawraga moisina neuroprotective property laibra haibado koina check tawjare aduda koina plant mapan tara mukti hoji pawda koina observe tawre adu process yam sang banna ko hoji purification tawdana pure compound yauna identify tawbodi ngamjadri adu makha tarakpa research ta adum honajarakini adu koi project sing se anambana di DBT ne DBT ne ICMR ne adagi IBSD ga collaborate tawba project masu yausaki ICMR project se dana koina DNA methylation na amalai dusu gene da tawba gene da methylation sina koi gi gene express tawba tawdaba sida effect lai halleba sisu koina drug addiction ga mari laina na check tawjabani adugi project se hoji tawri aduna hoji phawdi result to koi compile tawba loisadri masina punna oina koi project mari sigi major area of research ni haija tare
I am Nilkanta. I am a scholar in the University. I am a mi pung kuding mo koi DNA sequencing khena bai matung ina koi gi magi variation mayam do koina polymorphism mayam do koina check tawse bani so particularly koina drug metabolism gene da koina individual kuding mo ki koi gi magi gene sequence koina magi polymorphism do check tawraga koi gi population sida kama kang polymorphism na maximum yam na lei habi do koina check tawse bani polymorphism ji koina real time pisa thadar ganda koi gi magi maximum adugi minimum polymorphism do kone check tawraganda koi drug metabolism gene dugi ma effect to hoina either koi population jida yamna drug to number organda koi side effect yamna wang laigedra natragana thoidopa problem leiroidra habu koina mangjauna na particularly when tawje tuberculosis kini so tuberculosis drugs are amtong mangjauna na koina khangdokpa haitare side effect yam laigadra natragana koirom the side effect yamna thoidona leiroidra habu koina koigi nariba missing dugi matungina nariba missing plus nadriba missing dugi koina mangjauna na magi genetic koi polymorphism to check tawjabani
khoi hojik unajoi je khoi gi ngase khoi department asida khoi gi assistant professor oina pubire bi khoi gi dr sanatom bi gani ahan bada madam bu khoi gi Doordarshan Manipur na taranok seri
Thagachari ai Dr. K Sanatombi department asida assistant professor oina leizari ai 2009 degi department asida leizarakpani adu aina research tawjari ba area sina plant biotechnology ni ekoi Manipur University gi mateng amadi Government of India gi project kaya ga loina na khoi lab series ta thabak tawjabani akoina lokthajaba project mayam amadi university sigi mateng sina koina satra gi experiment kaya ama lab asida pangthokchari and maru oina akhoi gi lab asida akoina tawjari ba research aji plant biotechnology da plant cell culture haiba serum series da akhoi focus tawi akoina tawjaba se medicinal natragana aromatic plants hamna hairabda akhoi umrok kumba aduga shampoo ginger haina khangnari ba gingiber jiram bet kumba adagi kum haina phi sangbada sijina ba kum pambi gumba sugumba pambi gumba si akoina lab ta tissue culture tawraba matungda makhoi dagi akoina makhoi gi cell hairaga khangna ba kelas hana puthoi akoina arwagi puthoi ba kelas mayam sidagi akoina cell suspension natragana akoina moigi large scale culture si puthokna ba hotne aduna cell mayam si amadi kelas mayam si puthoga akoina pami ba si ngasai gi plant biotechnology gi application ama oina makoigi pambi sing sis da yauri ba kana ba potlam sing hairaga natragana akoigi secondary metabolites mayam se akoina extraction tawna ba protocol mayam ama sakatchabani aduna akoigi lab tagi akoina umorox ki akoigi manungi masingkha hairaga khangna ba moroki asaba part tu dagi akoina magi kelas puthokchare kelas puthokcharaga akoina kelas si masing yamna puthokchaba ngamjare aduna masugum basi akoina paper da phongjare aduga akoina puthoiba kelas sidagi akoina amuk cell suspension culture ana akoigi large scale da tawjare aduga hauji akoi project ama su taw chathajari akoina culture je akoigi scale up tawboina bioreactor da large scale da culture tawraga aduga akoina aim tawjari basi akoigi morok ta magi asa boina seasonary ba compound capsaicin haibase akoina karigumba morok tagi lothogadi chang yamna yaugdabanina morok mayam dagi khitang khak phangba ngami adubu akoina hojik aim tawjari besina cell mayam si culture tawraga morok ta depend tawdabida akoina hairiba potlam sing haibdi asaba natragana medicinal drug natragana dye compound sugumba compound mayam se akoina cell culture dagi lothokna ba akoina cell culture sing si tawjabani akoina tawjari bisi sumaina pambi singsi hana culture tawraga thajare si shampoo ginger hariba pambi duni sumaina culture tawre sina kum pambi ni akoina sumaina pambi singsi culture tawraga makhoi dagi mana machet kumba ama lauraga akoina sugumba kelas saina cell mayam se puthoi sugumba cell mayam se akoina masing yamna adum akoina tharaga makhal yam puthokpa ngamme asugumba mayam si leiraba matungda akoina makhoi si amuk large scale culture tawbagi damakta akoina sugumba bioreactor gumbada si benchtop bioreactor ni 1 liter volume da culture tawrini aduna 2 liter fawba tawbiyai aduga atopa hayeng kaigumba industrial scale da tawrokke hairagadi achoba bioreactor da culture tawrokku matamda sigumba cell mayam sidagi akoina pambi sida yauriba potlam sing haibadi morok ta hairaga magi asaba capsaicin kum da hairaga magi machugi compound indigo sigumba compound sing si akoina sigi cell sing sidagi direct lokthokpa yai aduna akoina ꯄꯥꯝꯕꯤꯗ ꯀꯔꯤꯒꯨꯝꯕ ꯄꯣꯠ ꯑꯃ ꯂꯧꯊꯣꯛꯅꯕꯗ ꯄꯥꯝꯕꯤꯗ ꯄꯤꯔꯤꯕ ꯄ꯭ꯔꯦꯁꯔꯁꯤꯡꯗꯨ ꯑꯩꯈꯣꯏꯅ ꯍꯟꯊꯍꯜꯂꯒ ꯄꯥꯝꯕꯤꯕꯨ ꯃꯌꯥꯝ ꯀꯛꯊꯠꯄ ꯑꯗꯨꯒ ꯑꯗꯨꯒꯨꯝꯕ ꯃꯌꯥꯝꯗꯨ ꯊꯤꯡꯅꯕꯒꯤꯗꯃꯛꯇ ꯑꯩꯈꯣꯏꯅ ꯁꯤꯅ ꯑꯜꯇꯔꯅꯦꯇꯤꯚ ꯁꯣꯔꯁ ꯑꯃ ꯑꯣꯏꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯃꯦꯗꯤꯁꯅꯦꯜ ꯅꯠꯇ꯭ꯔꯒꯅ ꯗ꯭ꯔꯒ ꯀꯝꯄꯥꯎꯟ ꯁꯤꯒꯨꯝꯕ ꯀꯝꯄꯥꯎꯟꯁꯤꯡꯁꯦ ꯁꯨꯃꯥꯏꯅ ꯑꯩꯈꯣꯏꯒꯤ ꯁꯦꯜ ꯀꯜꯆꯔ ꯃꯌꯥꯝꯁꯤꯗꯒꯤ ꯂꯧꯊꯣꯛꯄ ꯌꯥꯏ
ꯂꯣꯏꯁꯤꯜꯂꯛꯄꯗ ꯗꯤꯄꯥꯔꯠꯃꯦꯟꯠ ꯑꯁꯤꯗ ꯍꯦꯗ ꯑꯣꯏꯅ ꯄꯨꯕꯤꯔꯤꯕ ꯄ꯭ꯔꯣꯐꯦꯁꯔ ꯑꯥꯔ ꯚꯔꯊꯔꯥꯖꯟꯅ ꯁꯕꯖꯦꯛ ꯑꯁꯤꯅ ꯄꯤꯔꯤꯕ ꯀꯥꯟꯅꯕꯁꯤꯡꯒꯤ ꯃꯇꯥꯡꯗ ꯑꯁꯨꯝꯅ ꯊꯝꯕꯤꯔꯛꯈꯤ.
The students can get the job either in government or in private sector. In the government they can go for teaching program as well as for research program, either in the college or in the institutes or any other funding agencies like that. Under the private, again it is just like a C. They can go for private entrepreneurs. It is something like a self-employment. So if a student is able to get some fund from some bank, because many nationalized banks are giving money, with that objective they can initiate some business and they become self-dependent and they become self-entrepreneurs. This is one scope. If not, they can get into any private companies. Private companies are also coming up in large numbers. So there they can go for job as a scientist or a technical know-how persons or marketing. So like that a number of job opportunities are available. And the students after completing their post-graduation, they become research scientists. That is pursuing their PhD program or doing research in some research projects like that. Pharmaceutical industry is another big industry arena where the scope is enormous, because any product that you are previously the pharmaceutical units, they were synthesizing the medicines with the chemicals, with the raw chemicals. Resources are available, but now basic resources, chemical resources are reducing, so they have to go in for getting these medicines through biological means. And getting these medicine through biological means the biotechnological intervention becomes essential. So therefore pharmaceutical industry is very important for biotechnology students to get job. Then sometimes after doing their PhD they can become research associate. Government fellowships are available, a very good amount they are getting it with a lot of contingency and equipment purchase and so on. With that they can pursue their higher studies plus research associate and so on.
In between, sir, the importance of the benefits of doing JRF and SRF.
These days JRF and SRF, it is a good support. See, when after completing their MSc, a person who is writing the entrance examination with the JRF, CSE or background, he will get not less than 30,000 rupees per month with the contingency, and he becomes self-sufficient, self-relied for his financial. He need not depend on parents. So a student during the student life, per month getting 30,000 rupees is a good opportunity, a good amount. So with which he can buy the stationaries, then computers and other things, then establish his own lab, then have link with other collaborative people. So thereby he can expand. So it is a very good opportunity. First 2 years they are getting JRF, then third year after an assessment they are elevated to the rank of senior research fellow. We call it as a SRF. So from junior research fellow, after 2 years they become SRF. SRF means the fund is more, monthly amount is more. So this monthly emolument will make the student self-dependent. So no need to.
Then directly they can do PhD also.
It is simultaneously they can do PhD, and the JRF, SRF students they can also involve in teaching program with the department. So thereby during the period of 3-4 years of PhD duration, the student is not only doing research but involved in faculty research program and also taking care of the department in making or shaping the department in a better way. So therefore JRF and SRF, then DST has the Inspire program. So that also now included. A number of students have registered as the INSPIRE program. Actually, in Manipur University, the JRF as well as the INSPIRE program students, we are admitting them as a supernumerary. So it is like, for example, a teacher can take 6 students for PhD program. Means if someone comes saying that I am a JRF, I would like to do PhD, oh, you can join, no problem. So there the number of quota is not limited. They are considered as a supernumerary. So we encourage JRF and INSPIRE fellows. The opportunities are enormous. Then with their background in biotechnology, they become medical transcriptionist. And then they can also become food security officer, food safety officers. They can also involve in marketing. And also, of course, academic is always there. Academic, they are as a teacher, as a researcher, as a scientist, they can slowly expand their program. So I am just telling in nutshell about these biotechnological opportunities for students. But as a whole, when you look at biotechnology, we broadly categorize into 5 different types. Like biotechnology concerned with the environment, biotechnology concerned with the medical scenario, then agricultural biotechnology, then industrial biotechnology, then marine biotechnology. So these 5 important areas are there for biotechnology. So under the environmental biotechnology, we can go with a research program on pollution control, then recycling of the waste, and then producing renewable energy. So these are all important sectors wherein government is also ready to invest money. The researcher can also expand their research program in a very ambitious way. Whereas in the case of medical biotechnology, of course, medicine means always important for animal as well as a human being. So the medical diagnostics, a huge amount of investment is being made by the private sector. But if the student is able to make use of these diagnostic methods, then he can excel in his work. And if he is discovering a new method, then that method can also be patented. So that becomes an additional income, a kind of a bonus for the students. Not only for the student, it becomes an important discovery for the institute and also for the state. Now, drug production. Then these days, vaccine production becomes very, very important, which we realized it during the last 2-3 years when people had a COVID infection all over the world. So different pharmaceutical companies, they came out with a number of vaccines. So you buy my vaccine, you buy my vaccine, like that they were telling. It is all promoting their market. So at that juncture, if you are coming out with a number of vaccines, automatically our vaccine will be preferred by the government, our vaccine will be preferred by our own people. So thereby you get a lot of revenues. So vaccine production for animals and human being to prevent the diseases, very important, particularly for children. A number of vaccines. India is producing quite good amount of vaccines for children. So therefore the vaccine production becomes important, in which the biotechnologists play a vital role. And then like that, a number of healthcare areas are there where the role of biotechnologists are very vital. So similarly, agricultural biotechnology is very important because we are all living with the food crops. Okay, whether it is a paddy, wheat, maize, or any vegetables or fruits, we are all always interested in. And day by day, the cultivation area is getting reduced. So we want to expand. That means the horizontal expansion is not at all possible. We have to go for vertical expansion. So at that time, we need to improve the crop. So improve the crop means the crop production should also be more. At the same time, when you increase the yield, we cannot compromise with the quality. So quality should also be given maximum emphasis. So at that time, the biotechnology, agricultural biotechnology or plant biotechnology, they go for increasing the yield as well as increasing the quality. So thereby our products becomes better. So this is agricultural biotechnology. Now in addition to that, plants are always infected by a number of diseases, whether it is a bacterial disease or viral disease and so on, fungal diseases. So these diseases are rampant in many crops. So if you want to prevent the infection of these diseases, biotechnology can do a lot. They can modify the gene, thereby the plant becomes resistant, so that the plant will not be able to manifest the diseases like that of earlier. So from the susceptible plant, you are making a resistant plant through intervention and manipulation by genetic mechanism. So this is an important area, and the contributions are recorded a lot through plant biotechnologists, with the commitment and sincere effort of plant biotechnologists. Another important area under the agricultural biotechnology is, or general plant biotechnology is, conservation. Some of the plants are becoming less in number. I don't call it as a threatened species, but their number is gradually reducing, gradually dwindling. So in order to make their density, in order to make the population of these trees and plants more in number, the germplasm of the plants are taken, and through tissue culture process, more and more micro propagation is being achieved. Thereby the plant density is restored, so that we need not worry about the plant, whether will it reach endangered plant, will it reach a threatened plant. No such worry. Plant biotechnology can help us in that. Similarly, industrial biotechnology is another important area wherein students can concentrate on food technology. Okay, the food technology means enhancing the quality, enhancing the shelf life, and with least preservatives, the food can be preserved for quite a long period. And similarly, enzyme production is another important area under the industrial. So you may be knowing that synthetic chemicals are not now encouraged. So instead of synthetic chemicals, enzyme-based approach is involved. So suppose there is a stain in my shirt, so I need not bother about the chemical. With the help of enzyme, it can be removed. Okay, so that means it is a biologically accepted version. No pollution from the enzyme. Whereas if you are using the chemical, then chemical will pollute. So similarly, what they call the soft drinks or liquor production or fermentation technology with respect to rice and dal. These are all things where a lot of inputs are there, and slowly our biotechnology peoples are trying to tap the resources and trying to enhance the area, trying to concentrate more in production, more quality in production, so that the ever increasing population is fed with good amount of food, so that there is no shortage. We will have self-reliance in food production. Next is marine biotechnology. The moment I say marine, it is just like a ocean. You can a number of animals are there, a number of fishes or oyster or corals, like that. A number of animals are there, and these animals, including marine algae. So marine algae is one such a source, like a seaweed, spirulina. So from that we can get a lot of items. So health related vitamins and other nutritive materials can be obtained from the algae. And then algae technology has been introduced, and gradually it is gaining momentum in the algal research, and different laboratories in India are working with that. So students have particularly the botany students as well as the biotechnology students, they have a very good scope to enter into this region of algae technology, so that more viable product can be produced. Similarly, enhancing the fish production. So we always say that fish production is going day by day less. Then what is the reason? The reason is that the fishes are dying due to diseases or due to other environmental destruction, or you call it as a habitat destruction, and so on. So therefore, the fishes can be prevented from spreading or dissemination of the diseases through biotechnological processes. Simultaneously, fishes can also be increased in their production. So unit area, we can enhance the production. So thereby more and more fish means, it will solve at least to some extent our purpose, food purpose. So with the help of, in a particular area, fish production is more means through different remote sensing technique, they go to the spot, then identify the spot, capture the fishes in large numbers within short span of time, and bring back to the seashore and then sell it. So that will enhance the what they call common man's requirement as fish. So fish production leads to what they call the cost of production can also be reduced. People will not suffer due to shortage of fishes. So these kind of things. Then in addition to that, laboratories are involved in producing recombinant fishes. So thereby the recombinant fishes will be bigger in size, the biomass will be more. As a result, a family with 6-7 people, they need to buy just one fish and they can enjoy the fish for 2-3 days. So therefore fish recombinant technology has been now on the increase. And like that, then vaccine production is also important, especially for viral diseases which are infecting the fishes. So the scope is enormous. Now, in addition to our regular faculty members and students, we have also got a number of postdoctoral fellows in our department. And the postdoctoral fellows, their main aim is, after PhD, they can pursue research. So they contact the funding agencies, get some fund, a consolidated amount, maybe 50 lakhs, 30 lakhs, like that, they can bring. And then we are encouraging them to do research in our institute, in our department. So thereby they can take up some work in the field of biotechnology. So a number of students have registered with us, a number of students have participated in a collaborative research with us. And this collaborative research in conjunction with, in tune with our laboratory can yield more output, more turnover in our research program. And these are all some of the areas where we are concentrating. All the more we are interested in maintaining our quality. We are interested in going for patenting. We are interested in our name and fame automatically. Okay, once you do the technique and once you have patented it, automatically the methodology that you have patented will be the property of the Manipur University, and then thereby they will be able to get lot of income. So when a product is patented, so normally under the patent authorities of India, nearly for 20 years it will be on our name. So that within these 20 years, whoever is using that, so the income will be automatically coming to university. So this is one way of what they call generating fund for the department. So like that we are slowly expanding our wings, and I am sure that we will be able to achieve our goal over the period of time.